interest in any of these companies. And this is the agenda. I will try to start by the definition and give you some data on the epidemiology of the disease. And there are two different terms, POF, primary ovarian failure, and primary ovarian insufficiency, but we're talking of the same disease. The relevance of POI and why we study the genetics of this disease, and then we will go into the pathogenesis, and I will obviously talk about the relevance of genetics and give you the main genetics markers. And at the end, I will give you a proposal of how these studies can be useful for the management, clinical management of the families, and some future perspectives. So this is the distribution of the age at menopause in the general population, and you can see that the vast majority of women go into menopause around 50 years. There are few that go beyond, very few that go into menopause beyond 58, but there are several women that go into menopause before 45 years. And if we go look into the real number, you can see that 1 out of 100 women go into menopause before 40 years, and you can see that the prevalence of this disease decreased by a logarithmic exponential way but every decade almost. It is an heterogeneous disease. The most severe form are accompanied by primary amenorrhea. The less severe form occur in the fourth decade. Then there is another group of women that go into menopause between 40 and 45 years, and you can see that the number reaches very prevalent situation. 1 out of 10 women go into menopause before 45 years. And this has been in the past, let's say until 50, 60 years ago, not a relevant problem because the women used to conceive earlier during their life, but then the median age of conception is well now into the 30s, and so this problem has arisen, and this has become an interesting topic to study also. And why is early menopause so relevant? The prevalence, but then the consequences. Obviously, hormonal defect, we have some tools, the estrogen, to give to these women, and this can at least avoid most of the problems if you do it in the correct way and until the age of natural menopause in most of the women. But the most important issue is the fertility defect, which raises psychological consequences in the families, a lot of socioeconomical problems, and the only solution for these women at present is the oocyte donation. So, in order to improve and to avoid these problems and to improve the fertility or preserve the fertility of these women, we should make the diagnosis here. But you can see that here we don't have any marker, clinical or biochemical, that could suggest that this woman is predisposed to undergo this early end of her ovarian reserve. And usually the diagnosis is made here when the irregularity of the menses bring evidence of this problem, and you make the biochemical, and you see that the FSH is in the menopausal range, and you make the diagnosis. So if you think that in normal women, you see that 51 years is the median of median age of menopause, but subfertility starts 20 years before menopause. You should, in these women that undergo menopause in their 30s, you should, in principle, make the diagnosis very early during their life. And for this reason, there are several studies that are trying to identify markers, biochemical markers and genetic markers to diagnose these women in their early years. And for the biochemical markers, surely don't have time to go into details, but AMH is certainly the one that performs the best. There are several data showing that it correlates with ovarian reserve, and there are several studies that measure population in hundreds of population of women, the MH levels, women of different ages, and have constructed some algorithms to predict by measuring AMH let's say, at 30 years, and predict in some way the age at menopause of that woman. But in a single woman, it is very difficult to go earlier than 30 years. As you can see, this is the mean decrease in MAH levels that parallels the decrease in ovarian reserve, but this parallelism starts a few years before 30 years. So this is something perhaps for the future, but at present there is no possibility to do an early prediction of women at risk to develop POI. And since this is a recognized problem, there are several companies last year that, like Apple or Facebook, as I remember well, that offered to their staff women to freeze their eggs to preserve their fertility and offer the progression in their career. But this is very expensive and not available for all the women. So I think that a better understanding of the pathogenesis and to dissect what is the genetic predisposition to this disease would offer a possible solution. And these are the factors that are involved in POI. Obviously, environmental factors are very important. In first row, the chemotherapy and radiotherapy for oncological problems during in young women, exposition of toxics, and so on. And then there is the individual predisposition to this disease, and there are several factors that suggest that this genetic predisposition is important. I will not talk about autoimmunity because I don't have time to talk about it. But if you want, I will be available for some answer on this. The heritability of the age of menopause is very important. Probably the genetic component accounts for at least 50% of the inter-individual variability at the age of menopause. And women reporting a mother with an early age at menopause has a 6-fold increased risk of POF. So this is very important. And these are the models of POF transmission in familial cases in the Milan cohort that we have, and you can see that familiarity reaches 1 out of 3 women, and in the vast majority there is 72% that report a a mother that had the same problem or a similar problem. We have families that have primary, the proban is affected by primary amenorrhea with ovarian 46XX ovarian dysgenesis that has a mother that entered menopause between 40 and 45 or even earlier. So this is a disease with highly variable expressivity even within one family, and this may raise some ideas, but on the on the mechanism of pathogenicity, but then the maternal inheritance is a relevant fact. This may be obviously biased because the daughter can remember more easily the age of menopause of the mother and not that of the aunts or of the grandmothers, but then the numbers are very important, may suggest that X chromosome may have a role, and indeed this is the case. These are the defects that are associated with primary ovarian insufficiency in 46XX woman, or at least that do not had a previous diagnosis of Turner syndrome, and you can see that the X chromosome defects are among those with the highest prevalence. The monogenic disease, isolated disease, are less important in order of prevalence. There are many genes that have been associated, and there are also complex forms of the disease that may be recognized upon the phenotype in several cases. And so you could do, in principle, in these cases, do a directed diagnosis, investigation of the genes upon the phenotype, select upon the phenotype. Let's see what is the history. This is the Rosenfeld scheme of the gametes in the ovary. You can see that during embryogenesis, normal women have around 7 million of oocytes in their developing gonads, but then at birth there is a dramatic decrease of 1 million, and then the progressive decrease during the rest of their life, with the end at the menopause. And you can see here the recruitment of the antral follicles that starts at puberty. In most of the Turner women, we know that at least the model most recognized is that they lose their follicles early during development, and but there are some Turner women in which this decrease is somewhat conserved. There is a small percentage of follicles that is conserved, and some of these Turners can undergo a full spontaneous pubertal development, and they may even ovulate. And what are these Turner? Are those that have most likely a double dose of X chromosome there are mosaics for 45X, 46XX. You can see that in this case the FSH level are frequently within the low range, whereas in the classic Turner 45X, the FSH level are very high. And if you go and look for the subset of Turner that undergo the complete spontaneous puberty, there was an Italian study of some years ago that found that 9% of 45X could, at classic karyotype, conventional karyotype, could undergo pubertal development, and 33% of mosaics. And we recently performed this study with a more sensitive technique, the RACGH, and you can see that only one, in this case, 45X at RACGH could reach a spontaneous puberty. And I will come back to this case later. The implication of the X chromosome raised several the studies of breakpoints and highlighted several locus that are associated with primary ovarian insufficiency, and there are 3 locus that are associated now. The POF1 is FMR1, the gene that is very likely to be implicated in this locus. POF2 diaphanous is still a matter of understanding what is the role of these genes. That are no mutations so far reported in diaphanous. And then BMP15 is the 3rd gene that is localized in this locus on the short arm of the X chromosome. And obviously, FMR1 is very important. You all know that is the gene that is implicated in the Fragile X mental retardation, which is a relevant problem in boys, and is a disease associated to the expansion of triplets, CGG, and you can see this is the normal situation. In the 5' untranslated region of this gene, there are triplets that are less than 45. This is the normal situation. When you have more than 200 triplets in that region, there is complete silencing, methylation of this region, and complete silencing of the gene, and you have the Fragile X syndrome in males that inherit this expansion from the mother. And there are intermediate level of expansion that are called premutation that are associated with premature ovarian failure. And the FMR1 premutation is carried by 1 out of 250 women. Among women with the premutation, almost 1 out of 5 have POI, and about 11% of women with familial POI and 3% of those with sporadic POI carry the FMR1 premutation. And the earliest stage of menopause in Fragile X POI occurs in women with 80 to 100 repeats. The reason why this is unknown, but then they are rarely found under 20 years, so it's more likely to occur in late form of POI. And if you study FMR1 in a pedigree like this, you can see that these 3 women, the mother and the 2 daughters, had a FMR1 premutation, and if this investigation would have been done before the pregnancy of that woman, you could have predicted the possible risk of the birth of a boy with Fragile X syndrome. The other gene that I want to talk to you is BMP15. BMP15 is a member of TGF-beta superfamily. It's secreted by the oocyte in the follicular lumen, and it regulates several events in the granulosa cells. It inhibits FSH receptor expression. It reduces, it protects cells from apoptosis, and it stimulates proliferation of granulosa cells. And this is a member of this family, and the importance of this crosstalk between oocyte and granulosa cells become more and more important in recent years with the discovery of this feedback loop that involves several members of this family, with messages coming from the oocyte at the center and the others that are secreting various types of BMP from the theca and the granulosa cells. And the first possibility of implication of this gene in POI was given by our group more than 10 years ago. This is the family that we described with these two young women that inherited this mutation from the father. Both women had primary amenorrhea and ovarian dysgenesis without any stigmata of Turner syndrome. And we recently reviewed the implication of BMP15 in ovarian diseases, and you can see that by the analysis of several of the papers that after our first paper came out, once that we exclude some polymorphism, frequent polymorphism in this gene, you can see that the prevalence of variants, of heterozygous variant, non-synonymous heterozygous variation in POI is tenfold higher than in the control population, meaning that this may be a relevant susceptibility gene. And these mutations are mainly located in the pro region. There is only one so far reported within the mature peptide. The pro region is very important for dimerization and maturation and for the secretion of the protein as a dimer. There are several mutations that have been investigated in vitro and others that do not, so we still don't have the entire picture understood. But we recently identified a POI case with primary amenorrhea that is carrying a biallelic deletion of the only BMP15 region on the X chromosome as determined by array CGH. So this is another proof of the possible role of this gene. In several cases we demonstrated the lack of the diminished production or in vitro of the mutant proteins, and this may in some way be the role of BMP15 of these variants would be that the decreased amount of BMP secreted into a follicular lumen may favor the apoptosis of granulosa cell and the atresia of the follicle. So when we published one of the first papers on this topic, there was an editorial that was written by layman that said, maybe this is the first true ovarian determinant gene on the X chromosome, and we have some evidence now that may say that probably, perhaps, yes, this is relevant for ovarian development. And I can remind you again the case that I told you before, the Turner patient that had a 45X conventional karyotype but had spontaneous pubertal development. She presented at the age of 11 years for growth retardation and was put on GH treatment, so she was recognized as Turner at 11 years. The present height is, however, small. She was born at term. She had no sign of Turner syndrome. She had a spontaneous menarche at 14 years, followed by regular menses, but amenorrhea of 3 months at 18 years with these levels of gonadotropins was found at 18 years and was put on treatment, obviously. She had no pregnancy, but then we made the array CGH analysis, and you can see that we could detect a double signal for the X chromosome by array CGH only on a portion that contains only BMP15 as the only entire gene contained in this duplication. By fish, we demonstrated that this is a tandem duplication of the BMP15 gene. So this Turner patient is 45X, is really 45X, but has a double dose of BMP15, suggesting that a double dose of BMP15 appears sufficient for complete spontaneous puberty in Turner 45X. We would like to find more, but this is apparently rare. But these evidences that we brought about brought some other researchers to evaluate the levels of BMP15 in the follicular liquid and to predict the possible oocyte that may be of higher quality in poor responders to ovarian hyperstimulation. Another gene that we are now studying in collaboration with Nadine Binard in France is NOBOX, is a transcription factor that is known to stimulate the production of GDF9, that is the partner of BMP15, and we found mutation in 4.3% of this cohort of women, of 490 Caucasian and African women. And you can see that the studies that we are doing show that these variants, they can be expressed, but they do not go into the nucleus as the wild type does. Here you can see that there is little amount of mutant proteins that go into the nuclei of the cells. So why these studies may be useful? If obviously we cannot make the diagnosis of this disease. The diagnosis is still at present because it is still done at the stage of amenorrhea in the proband of these families. Then we suggest that a complete clinical characterization, I mean to suggest maybe an autoimmune origin of the disease, or more likely a genetic origin of the disease. You can in first row or investigate autoimmune biomarkers, or then make the first-line genetic test, which may be in POI before 25 years, do the karyotype because of the possibility of hidden Turner, and above 20 years of age, obviously, also FMR1 becomes important. And then there are specific genes upon phenotype. I don't have the time to talk about this. In the next future, I think that NGS analysis of candidate genes or whole exome will become, obviously, more efficient for this diagnosis. So this is the final picture at present of the prevalence of all the variants that are under study in our lab. And we think that by the NGS of array for genetic counseling in families at risk, that we at present can make a diagnosis in 30, at least found a susceptibility gene in 30% of the cases, we can then go and make an early diagnosis and cryopreserve or preserve the fertility in the young relatives of this woman, and obviously obtain the informed risk of X-linked mental retardation in these families. Future perspectives, very few words if I have the time. Do I? Okay. We are trying to expand the knowledge and find new candidates by a study of the expression of the transcriptome in granulosa cell upon BMP15 stimulation, and we recently obtained evidence that BMP15 acts as a repressor, mainly as a repressor of transcription, and we identify novel candidate that we are now analyzing for the pathogenesis of this disease. And then there is also the problem of mitochondria, which are also inherited from the mother. And you all know that oocytes are cells that are very enriched in mitochondria. They are the second after heart cells in the body. And you can see that there is this study showing that mitochondria content is low in oocytes from poor responders and associated with ovarian insufficiency. And we studied the correlation between blood mitochondrial DNA content and ovarian tissue, and we found that there is some correlation. So we went to the blood of old women and control women, normal responders to stimulation, so of the same age of the POF or the poor responder women, and we found that the mitochondrial DNA content in the women with ovarian reserve is significantly lower than in the very old control with physiological menopause and women with intact ovarian reserve. So maybe future studies will tell us if blood mitochondrial DNA determination may become a supplementary useful biomarker in the prediction of the risk of premature ovarian failure. That's it. I want to thank the people that contributed to this study, the people that work in my lab, all the collaborators, the clinicians, and the scientists that collaborate with us on these studies, and the funding bodies. Thank you.
Open for question and answer. Maybe I ask a question about the Turner syndrome. We know a portion of the X chromosome could be inherited from father versus mother. I don't know if there's enough large study shows the differential inheritance of the X chromosome from father versus mother. Is there any correlation with the ovarian insufficiency?
I don't know. There is no such data.
And for the other genes that are not associated with the X chromosome, how many do you think there are out there? I mean,
there are a lot. I showed you a picture. You all remember the story of the FSH receptor that was prevalent in the Finnish population, but then looking at other population, this prevalence was very low. In our setting, we found 1 out of 400 women that was carrying a double heterozygosity, compound heterozygosity for FSHR gene. There are genes that are susceptible to be frequent. One, the autosomal gene is no box, but then there are other transcription factors like FOXL2 or that maybe are important and more important than presently known. So there are several things that needs to be done. I must say that since there is a high I mentioned the highly variable expressivity of the disease with the age of the onset of menopause in the same family, in different women of the same family. There are some data that are coming soon, will come out on the multigenic origin also of the disease. So that's why I say susceptibility. When you find something, you cannot say that this is the only cause of the disease, but this is an interesting approach and for the interesting issue for the novel approaches with like NGS approach for discovering this kind of multigenic involvement.
Do you also find differences between ethnic populations, let's say from Africa, from Asia and so on? Is there some evidence that the prevalence is different? And can you also give some explanation to that with respect to the genes?
This is very difficult, but there are differences in populations. When as far as BMP15 is concerned, some SNPs are more frequent in the black population than in other population. The Japanese population, for example, do not have these SNPs, and it was found to be, the women from Japan were found to be negative for BMP15 variants so far. But I don't have other explanation for this. The origin, yes.
Yes. No, okay, thank you.
Population genetics.
Yes,
and also for diaphanous, there are some SNPs that have been associated, but also for inhibin, that are SNPs that have been associated with different involvement in different population. But the definitive role of this SNP is quite very difficult to...
Can you speculate on the relative importance of some synergism between environmental exposures and maybe the genetic background, because that might be also the reason
this is a
for...
very important thing. Also for the young women that undergo toxic treatments like the chemotherapy, because if the chemotherapy is given to women that already have a diminished reserve, they would have a more severe impairment of their reserve after the chemotherapy. So this is also a problem to address, also to investigate in the population that undergoes this kind of treatments. And I don't know if predisposition to tumors may also predispose to ovarian insufficiency, but this is another story.
Yes, I would have guessed that you find more genes also in meiosis, but maybe they are selected.
There are new candidates in the synaptonemal complex.
Okay, thank you.
May I ask a last question about back to this Fragile X syndrome, the premutation? You mentioned like roughly 20-30% patient.
I don't understand.
Let me repeat the question about the Fragile X mutation. In the premutation, you mentioned roughly 20-30% patient, or 100%. I missed the among the patient that carry the premutation, like the repeats is 55 between 100. There are how many patients? What's the percentage of patients that show the ovarian insufficiency?
Among the FMR1 carriers, the premutation carriers, is around, as I told you, is around 20%.
Yeah. Why is there so many? Like, what are the other 80% do not show?
Sorry. The rest may be that since there are different kind of expansions, and there is also the possibility of the multigenic effect, so there are other genes or other environmental factors that may become important additional factors that together with the permutation cause the primary ovarian insufficiency.
Okay, thank you very much.